Hi, this is Miss Mansour, and in this video, I'll be talking about what mitosis is, how it's regulated, and the internal and external factors that allow cells to divide. This video is important because the loss of the control in the cell cycle can lead to cancer. So, let's get started. Now, before we get going, we first need to know what mitosis is. Mitosis is a type of cell division that results in two identical daughter cells. These cells are used to replace broken or dead body cells throughout the body. Now, the first step of mitosis is interphase, and this is the longest part of the cycle and can take up to 18 to 20 hours before it finishes. This phase is subdivided into the G1 phase, the S phase, and the G2 phase. But we're not going to worry about this right now. Instead, we're going to worry about how the cell goes through mitosis and the cell cycle. So, as we said, the first part of mitosis is interphase. Now, basically, what interphase is it's when the cell is preparing itself to go through cell division. It's growing, it's preparing itself, it's going to replicate its DNA. So basically, it's why it's the longest part. But just remember, during interphase, nothing is being divided yet. Now, the second part of the cell cycle is a mitotic phase, and in the mitotic phase, it consists of prophase, prometaphase, metaphase, anaphase, and telophase. So, during prophase, this is when the chromosomes become visible as paired chromatids. So, as we can see here, the chromosomes here are just tangled up, and if you were to put this under a microscope, you wouldn't be able to see the chromosomes. So during prophase, the chromosomes are being condensed and coiled so that they can look like this. And if we were to put this part of the cell under the microscope, we would be able to see the chromosomes then. And these chromosomes are called sister chromatids. So then the next part of the cell cycle is prometaphase. And during prometaphase, this is when the nuclear envelope disperses. So as we can see in prophase, we still have a nuclear envelope, but during prometaphase, this nuclear envelope starts to disperse and disappear. What also occurs during prometaphase is that the chromosomes start to connect to the spindle fibers. So as we can see here, these are the spindle fibers, which are microtubules, and they're connected to the centromere here, and then they're connected to the chromosome here. So after prometaphase is metaphase, and this is when the chromosomes have reached their maximum condensation. And as we can see here, the chromosome is condensed, and so they line up at the metaphase plate, and they're still attached to the spindle fibers, and they're getting ready to divide, but they haven't divided yet, which division occurs during anaphase. Anaphase is when the chromosomes are pulled apart, as you can see here. They're being pulled apart by the spindle fibers, and they're going the spindle fibers into two daughter chromosomes. These chromosomes move to the opposite sides of the cell. And then the last phase is telophase, and this is when the chromosomes go back to chromatin form, and also the nuclear envelope reassembles. So as we can see here, we had a chromosome here, but then once telophase happens, it goes back to its chromatin form, and the nuclear envelope is being reassembled. So now that we know what mitosis is, we need to know what the subphases of interphase. As we said, we have the G1 phase, the S phase, and the G2 phase. So as we said, interphase is when the cell is getting itself prepared to divide. So during the G1 phase, the cell is preparing itself to divide, it's growing, it's getting bigger, and it's just preparing itself to divide. But during the S phase, this is when the cell starts to replicate its DNA. But while it's replicating its DNA, it's still getting bigger, and it's still growing, and it's still preparing itself. And then during G2 phase, it's still growing, it's still getting bigger. It's just making sure that everything's ready before it goes into the mitotic phase. Now, as we said, mitotic phase has subdivisions of prophase, prometaphase, anaphase, and telophase. And we also have, during the cell cycle, we have checkpoints. So we have 3 checkpoints in the cell cycle. The first checkpoint is at the end of the first phase in interphase, which is the G1 phase. And this basically checks to make sure that everything went well in the first phase and make sure that the cell can continue on to the S phase and the G2 phase. The next checkpoint is at the end of the G2 phase. This checkpoint makes sure that interphase happens successfully in order for the cell to enter the mitotic phase. And basically just make sure that the DNA replicated correctly and that this cell is ready to go into the mitotic phase, because once it enters the mitotic phase, the anaphase, there's no going back. Anyways, so, and then the last and third checkpoint is at the end of the mitotic phase, and it makes sure that the daughter cells are okay and that they're able to go through the cycle again. Now, how does a cell know when it can go through the cycle, when it can't go through the cycle, and just when does it know when to go through the cycle? So basically, internal and external signals that provide a stop and go sign. So in order for a cell to know when to go through the cell cycle, there has to be a certain amount of proteins accumulated that signals a cell to allow it to go through the cell cycle. These proteins are called cyclins. Now, these cyclins are proteins associated with cell division and the accumulation of cyclin, and the accumulation of cyclin initiates the start of cell division. CDK, which is cyclin-dependent kinases, are proteins that are activated when combined with cyclins and are involved in regulating the cell cycle. So here we can see the CDK, here we can see the cyclin, but let's remember, the cyclin-dependent kinase, that means the CDK can't act on its own. It needs the cyclin in order for it to act. So when the cyclin and the CDK bind together, they create a complex called MPF. And MPF, it stands for mitosis promoting factor. And basically, this helps regulate the cell cycle, and it allows cells to enter the cell cycle and divide. So that's one of the signals and stimulations that allows the cell to go through this. So the next part, as we can see here, is the MPF. So what is cyclin and CDK? As we said, cyclin is a protein, CDK is basically an enzyme. So here's a cyclin, it's the substrate, here's CDK, it's an enzyme, and they bind together at the active site to create the complex called MPF, which is the mitosis promoting factor, which again regulates the cell cycle. Now, what is PDGF? This is another stimulation that allows the cell to go through the cell cycle, and PDGF stands for platelet-derived growth factor. PDGF is required for the division of fibroblasts in culture. Fibroblast is a type of connective tissue, and the binding of PDGF molecules to these receptors triggers a signal transduction that allows the cell to pass the G1 checkpoint and divide. This picture here shows an experiment of fibroblasts. This is a type of connective tissue, as I said before, and so when an injury occurs, these platelets release PDGF, and this results in proliferation of fibroblasts that helps heal the wound. So, how does cancer occur? Cancer, well, let's first start with this. DNA controls the actions of a cell, and one of the important functions is how the cell makes a copy of itself. So the cause of cancer is when there's a damage on the DNA, and that's called a mutation. So when the DNA has a mutation, it could be a cancer cell. So a cancer cell will divide over and over and over again, and eventually this creates a tumor. So as we can see here from this picture, we have a bunch of normal cells here, and as we said, normal cells are only replicated, only made when a cell is dead or is just deformed or denatured or whatever it is. But as you can see here, there's a big blob of cells here, and these are the cancer cells. They just keep replicating over and over and over again. They don't ever know when to stop because there's a mutation to their DNA. So it causes an overcrowding and overgrowth of cells. So as we can see here, this is breast cancer. So if we look at the normal breast, we can see here that there's a normal amount of cells and it's very normal and there's nothing wrong with it. But if we look over here, we can see these tumors, and as we said, tumors is due to the overgrowth of cells. And the only way to stop cancer cells from growing is chemotherapy, and that's because chemotherapy stops DNA replication, cell division, and blood vessel growth. So that is why cell regulation is very important, because if cells weren't regulated, cells would just keep replicating over and over again, and this would cause cancer. And I hope that helped, and have a great day.